of the most important players in these cell death processes. I'll try to explain that to you. Later on, well, in historical view, later on in the 90s, part 1 knockout mice were constructed, and then it was discovered that part 1 also plays a role in signaling of well, redox-related processes, the induction of inflammation, and that's another way. It also mediates reactive oxygen species production and its effects. And to complete the whole picture, I will show you a little on how this enzyme is also related to aging and aging-related diseases. So, cell death is the first topic. You may also already know that we have different types of cell death: apoptosis, necrosis. So, apoptosis is considered a clean form of cell death. The cell shrinks, the chromatin condensates. You have this budding, this blubbing of the membrane, particles that go out of this cell and the apoptotic bodies. These are phagocytosed by the surrounding tissue cells, and usually there's no inflammation. Usually. Necrosis, the other type of cell death, is thought to be a very damaging process. Cell membranes become leaky, contents leak out, and the cell shrinks and is lysed, and this leads to inflammation. That's the classical view. Necrosis is thought to be due to stress and due to damage to the cell. Apoptosis can be divided in two types of apoptosis: intrinsic and extrinsic. And I'll show you a little more about later. Important features are that for necrosis, you see that the cells get depleted of ATP. The loss of contents is into the environment, and this is a very strong trigger for inflammation. Apoptosis is an ATP-dependent process, so it requires energy to be performed. It's mediated by caspases, and there's no or only mild inflammation. So I hope this one is visible because the light is still. Can I do something about the light? No?
Als je een knop VGTL bij.
This one? Is it better like this? So, if you have apoptosis, there's extrinsic and intrinsic. Intrinsic is mediated by the FAS death receptor. So that's binding of a ligand to this receptor, and then apoptosis signaling occurs, which results in caspase A activation, which activates caspase 3, which leads to the execution of apoptosis. The extrinsic form is mediated by, among others, reactive oxygen species and other kinds of stress, which leads to usually an effect on the mitochondria. The membrane potential will drop. Mitochondria will release cytochrome C. There is some anti-apoptotic signaling, but if that is not sufficient, this will also continue in caspase 9 activation, which activates finally caspase 3 and will lead to cell death. So, in short, this is apoptosis. There's little more to tell about cell death. So, the first one, this is the caspase 3 mediated apoptosis. This is mediated by the death receptor, caspase 8. This is one. I just explained that. But we also have some other forms of cell death, which are induced by stress and which are mediated by other proteins. So in this picture you can see that also calcium is one of it. It's a little strange. Here calcium release, intracellular calcium concentration rise, and this also leads to a membrane potential drop of the mitochondria to ATP depletion, and this is a caspase independent form of cell death. This is the more or less the classical necrosis. But there's another one which is also mediated by the mitochondria and which involves either cytochrome C release or apoptosis inducing factor release. So these two are also caspase independent and also lead to cell death, but it's not really the classical form of apoptosis. So we have now a different separation of cell death processes, which is into caspase dependent or caspase independent cell death. In this case, you can really see it's programmed, so there is signaling involved. There's a complete caspase cascade. You see this typical laddering of DNA into 200 base pair fragments, and it's ATP dependent. The caspase independent can be both programmed, so what I showed you with the apoptosis inducing factor, or non-programmed, which is the classical type of necrosis. Caspase can be involved, so it's not sufficient to say, well, I don't see caspase activity, so it should be caspase independent. No, there can be involved, but they are more like secondary players in this cell death process. You see laddering of DNA into much larger fragments, so either 5,000 or 50,000 or even larger fragments, and that's because an endonuclease is involved, which fragments this DNA into much larger fragments. This process is not dependent on ATP. And the secondary role of the caspase is that these caspase later on can cleave these fragments as long as there's ATP present. Okay, so how can we follow this kind of cell death processes? Well, one of these assays to measure DNA fragmentation is the common assay. Most master's students will already know this. I don't know how others, if they're familiar with this assay, but in general you have your cells, they have been treated with an agent, in this case reactive oxygen species for instance, and you put them in an agarose layer on an object slide. So this is a microscope slide with agarose on it, and here the cells are. They will be lysed first, so lysis means membrane lipids will be removed, proteins will be hydrolyzed, so you end up with just the nucleus in the agarose, so only DNA is left. Then you will put the slides in a high alkali solution, and this at a pH 13 or higher. This will lead to denaturing of the DNA, which means that the double-stranded DNA will become single-stranded, specifically where the fragments, where the breaks in the strands are. Then you will do an electrophoresis. DNA is negatively charged, so it will move to the positive pole, and then you will stain with a fluorescent dye, and finally under fluorescent microscopy you will see this kind of images. And you can also quantify the amount of DNA strand breaks. This is a typical picture of untreated cells, treated with 50 micromolars hydrogen peroxide, 100 and 500. And this is typically a picture you see of cells that are apoptotic or necrotic. So this is a dying cell. This will not survive. So you can really see that oxidative stress, and in this case it's again hydrogen peroxide, will lead to cell death. Here you can see the cells were treated, and on this y-axis the ATP levels are shown. You can see them clearly drop. On this axis the LDH, lactate dehydrogenase leakage, which is another measure of cell death that's rising after treatment. So this is clearly a normal picture of measurements you can do to study cell death. But the classical view is also that hydrogen peroxide, which leads to the formation of hydroxyl radicals, induces cell death, and that you can reduce this cell death or inhibit this cell death with antioxidants. But that's not what we will talk about today. This is also a way. Inhibition of the poly-ADP ribose polymerization preserves the glutathione pool, so reduces the oxidative intracellular oxidative stress and reverses cytotoxicity in hydrogen peroxide treated lymphocytes. So not only antioxidants protect against hydrogen peroxide induced cell death. So this PARP1, what does it do? It's an enzyme that is first discovered in 1966, and it's later on other members of this family were also discovered, and at this moment we have some 18 proteins which belong to this PARP family. It is also called, at least it was proposed a few years ago, but hardly any scientist follows this proposal, to call it ARTD1, which means ADP ribose transferase diphtheria toxin-like 1. Okay. Almost everyone calls them states PARP1. This is the general building of this enzyme. So here is the catalytic domain. This is also called the domain with the PARP signature. So all the PARP family members have this domain. This is where the enzyme binds NAD+. Here's a DNA binding domain, which is typical for PARP1, for instance, but also PARP2 is a DNA, has a binding domain. It has 2 zinc fingers. It has a nuclear delocalization signal and some other proteins. And here is, in the middle, is the auto-modification domain, which may sound a little odd. Auto-modification means it can modify itself. This is, and I'll explain later, this is also a domain where it interacts with other proteins. So other proteins can either connect or bind covalently to this domain. So here you have an overview of the complete family of PARP enzymes. You don't need to know this. What is important is that this is the PARP signature, so you can see this. And PARP1, PARP2 are important as long as I'm talking about cell death. And also the tenkirase 1 and tenkirase 2, these two have some meaning later on in this lecture. So what does PARP1 do? Well, it has enzyme activity, and specifically the induction of DNA strand breaks is a very strong trigger for PARP1 to become active. So oxygen free radicals, gamma rays, X-rays, also alkylating agents. So these are agents that either methylate or acetylate bases and in this way induce modified DNA bases and induce DNA repair. Bleomycin, which is an intercalating agent, they all finally induce strand breaks, and this is a very strong activator for PARP1. What it does, it catalyzes the hydrolysis of NAD+ into nicotinamide an ADP ribose. And this ADP ribose is attached to a glutamine group of a protein acceptor. And it doesn't do this one time, no, it forms polymers. These can be linear, these can also be branched. And the effect of this process is that PARP activity requires a lot of NAD+. And NAD+, you know already, is also very much connected to ATP synthesis. And here's the clue to the cell death process. So after induction of strand breaks, up to a 500-fold increase in activity has been measured. So what's next going on? Well, PARP is activated by the DNA strand breaks, but it's not a single molecule. No, usually it forms a dimer. So two molecules of PARP bind to this region where there is this strand break. It can also be PARP1 plus PARP2. So here PARP2 comes in. Then it starts synthesizing these polymers. But these polymers are very negatively charged. So the ADP ribose polymers have a very negative charge. DNA is also negatively charged. So what happens is that when there's a lot of polymers synthesized, this molecule will dissociate from the DNA. So and then the polymers are degraded by an enzyme that's called PARG, poly-ADP ribose glycohydrolase. So these polymers will be removed, PARP molecules are in their native form again, ready to be active again. So this is what's called the shuttling of PARP1. Why does it do this? So its main function is to enhance the repair of DNA strand breaks. So it has been found that if you have knockdown or with siRNA removal of all your PARP, DNA repair process of single-strand breaks, double-strand, are not proceeding very fast. So at a very, very, very low level, too low for cells to be able to cope with DNA damage. So here you can see that there is DNA damage, PARP binds, it becomes active at this site, and it synthesizes its polymers. Repair enzymes are recruited. So the main function of PARP1 is to recruit the DNA repair enzymes that are necessary, and for these enzymes to be able to perform their task at the right place, there has to be space. So the synthesis of these polymers will also lead to a loosening of the chromatin. So at this place, the chromatin structure is loosened. The enzymes can reach the place, and well, PARP is also negatively charged when polymers are attached, so it will detach, and the enzymes will do their job, and you will end up with a repaired DNA strand break. What you can see or measure of PARP activity is, well, just follow the levels of NAD+ in cells. That's what we did like 12 years ago when we started to study PARP1. So here you see we have had some lung cancer cells, the A549s, treated with hydrogen peroxide. This is 100 micromolars, 300, and you see a nice drop in NAD+ levels. When we added the 3-aminobenzamide to the 300 micromolars, we could almost completely prevent this NAD+ drop. So this is basically what you do when you want to know is PARP activity involved. You follow the intracellular NAD+. So how about NAD+? Well, NAD+, and this is a simple picture, you should know this. NAD+, it's used here by PARP1, and under normal conditions, PARP1 is also active, but not at a very high level. So NAD is used, nicotinamide is released, these polymers are produced at a very low level. Nicotinamide can be recycled back to NAD+, but that requires 4 molecules of ATP in total. So to regenerate NAD+, it costs a cell 4 molecules of ATP. NAD+ is usually necessary for the glycolysis. So here you see glucose converted into pyruvate. NAD is reduced to NADH. NADH is in the mitochondria, is also produced in the citric acid cycle, and the electrons via the electron transport chain finally produce or lead to the production of ATP. This ATP is also used here in a part of the glycolysis. So this is ATP production, or it's produced here, and it's tightly connected to the NAD levels. At high levels of PARP activity, so if cells are exposed to high levels of reactive oxygen species, the polymers are produced at a very high level. So a lot of NAD+ is now entering the nucleus. This NAD+ is no longer available for glycolysis, which means that there is also less NAD+ available for the electron transport chain. Also, NAD is leaving the mitochondria. But mitochondria also require ATP to maintain their membrane potential. So the overactivation, as this is called, of ATP, of PARP1, can lead to an energy crisis. Well, these polymers have also a very specific role, it is thought. So that's the topic of your paper for next Friday. When they are massively synthesized, the glycohydrolase is not as efficient as the production of these polymers. So these polymers, they also leave the nucleus, and they are thought to be a cell death signal as well. So cell death can be induced here by 2 mechanisms. It can either be the massive drop in NAD+ levels, which leads to a reduced or at least impaired ATP production, and if this goes on for a longer time, even ATPase activity will occur here, so not ATP produced, but it will be cleaved. So the cell will enter an energy crisis, which leads to cell death. But the other alternative is that these polymers are a specific signal to the mitochondria that leads to cell death. So this cell death pathway, mediated by PARP1 activity, has been postulated to have specific function. Because if there is massive DNA damage and a cell doesn't have enough energy to repair all these DNA strand breaks, the cell will enter a cell death or will go into cell death. And this may prevent the cell from surviving and proliferating with unrepaired damage. So it will prevent the survival of cells with unstable genomes. And that is, of course, one of the ways cancer cells may evolve. So this cell death is thought to be, a beneficial thing to prevent the survival of genomic unstable cells. Okay? So there are different cell death procedures and also PARP mediated, and that depends on the types of cells. First, this panel. In the post-mitotic cells, so that means in tissue cells like cardiac tissue, kidney tissue, brain tissue, neurons, these usually have a low NAD content. And they are also heavily dependent on oxidative metabolism. When there is massive DNA damage, DNA strand breaks, and for instance, by one of these processes, ischemia, reperfusion, stroke in brain, septic shock, cells will have PARP overactivation, and this will lead to the cell death process that involves either apoptosis-inducing factor or is mediated by this NAD drop. This will lead to inflammation and to tissue injury. This process has different names. If you look it up in the literature, you will find different names. Chromatinolysis is the classical name, but since the PARP polymers have been identified to play a role as well, it's also called PARP thanatos. So this is what happens in post-mitotic tissues. A PARP inhibitor can reduce this massive drop in NAD+, or can reduce the massive formation of these polymers. And this will prevent this process of cell death, and has also been shown to be one of the treatment options to prevent the damaging effects of ischemia reperfusion. The other situation is where you have tumor cells, or cells that are very much replicated. Even if there is limited DNA, and when there is limited DNA damage, in this case we are talking of a different situation, you can have part 1 and part 2 activation, and usually this leads to DNA repair, cell cycle arrest, and survival. But if you use a PARP inhibitor here, the DNA strand breaks will not be repaired, or at a very, very low rate. This means that the cell cycle arrest will continue. And that also means that you will have p53 activation and p53 mediated apoptosis. And that is one way in which you can use this PARP inhibition as an anti-tumor therapy. So in tumor cells, a little too much. In tumor cells, you can use PARP inhibitors to kill your tumor cells. So, recapitulating what happens when you have PARP overactivation and cell death. Here is the trigger. So you have either high levels of oxidative stress, an anti-tumor treatment, something that induces DNA strand breaks. So you can see the level of the ADP ribose polymers rise until a maximum is reached, and then they will reduce again because glycohydrolase is also active. But if there's sufficient ATP left, you will also have cell cycle arrest, but also the induction of p53 mediated apoptosis. So your caspase activity will rise. And PARP1 is one of the major targets, substrates of caspase 3. So, and that's also connected to this, cellular energy metabolism function, because as long as PARP1 is active, it will consume NAD+, it will also consume ATP. So there's no ATP to perform your caspase-mediated cell death. So one of the first things caspase is cleaving or hydrolyzing is the PARP1. So you will have PARP1 fragments. And that's a very much used measure to determine apoptosis. So those of you who have done some apoptosis studies or have read some literature will know PARP cleavage is one of these measurements you do to study apoptosis. If you have cleaved PARP, then there is caspase activity, so you have apoptosis. But if there is not enough ATP left, you will also have this apoptosis-inducing factor release and some endonuclease activity. So these are all processes that occur if you treat cells with high levels of DNA damaging agents. So recapitulating, DNA strand breaks, if they are not repairable, you will have a massive drop of the NAD+, but you will also have unrepaired DNA damage, which leads to caspase activation, PARP will be cleaved, and you will have apoptosis. This is the situation where you will have normal repair, normal PARP, well, at least PARP activation at a level that a cell can cope with, and you will have cell recovery. And this is what happens if there is so much DNA damage that your NAD and ATP levels drop, and this is then called necrosis. In this case, there is no AIF, but AIF is somewhere in between these two. There's another thing to tell you about the anti-tumor therapy, and that is that one of the major applications of PARP inhibitors has been the last years the anti-tumor therapy. And specifically for these tumors that are deficient in BRCA1 and BRCA2, so breast tumors usually, that have no BRCA1 and BRCA2. And I'll explain you a little on how this concept, how it works. So anti-tumor agents, the usual ones, they induce DNA damage. So it's either radiotherapy, alkylating agents, induce DNA strand breaks. If you have cells that are not dividing, PARP will become active. It will start synthesizing its polymers, and if these polymers, if this cell has enough NAD and DNA repair is induced, cells will survive. But that's not what we want. We want cells not to survive, so that's why we use PARP inhibitors. So with PARP inhibitors, DNA strand breaks will not be repaired. If a cell starts proliferating, at the replication fork, there will be a gap, which means that the replication fork will be stalled. And that's also the case in this situation. If the cell is dividing, there is this strand break, there will be a stalled replication fork. Now, PARP1 is also part of the double strand break repair pathways, like homologous recombination and non-homologous end joining. It's not necessary to exactly know what these do, but these are specifically pathways that are involved in repair of double strand breaks. So, so if you have a PARP inhibitor here, cells will not be able to perform the homologous recombination or non-homologous end joining. And these double strand breaks will continue to exist, and that is a very strong apoptotic signal for cells. And this double strand break repair usually also involves the BRCA1 and the BRCA2, and that's the reason why in BRCA2, BRCA1 deficient tumors, this is also a very successful therapy. So this application of PARP inhibitors is at this moment really tested in clinical trials. Another important feature where PARP1 plays a role is in ischemia reperfusion. And that's the induction of inflammation. So I told you already with the discovery of part 1 knockout mice, or construction of these mice, that was somewhere late in the 90s, previous century. It was found that these knockout mice were highly resistant against endotoxic shock. So what is endotoxic shock? You just inject LPS, which is a bacterial cell wall component, intraperitoneally in the mice. And they will develop a very massive septic shock, which normal mice, they don't survive. So you can see here, all 10 normal mice died, wild types. But the part 1 knockouts, 9 of 10 survive. What's the reason for this? Well, they found out that the part 1 knockouts, they didn't have NF-kappa B activation. So they couldn't detect NF-kappa B activation. And they also couldn't detect IMOS. Whereas in the wild types, there was a clear induction of IMOS. So it means that part 1 is also necessary as a, what we now know, co-activator for NF-kappa B. So how should we see this? Well, if you have this LPS, this will lead to NF-kappa B activation, so it will be released from this inhibiting protein, it will travel to the nucleus and will start mediating the transcription of these NF-kappa B mediated GA genes, which involve the pro-inflammatory cytokines IL-1, IL-6, TNF-alpha, and IMOS. So these will start producing, well, IMOS will start producing NO. There's also superoxide and oxygen radicals, and peroxynitride will be formed. This will lead to DNA damage, to PARP overactivation, well, and in the end to cell death, necrosis, and systemic inflammation, and this is what happens with septic shock. So with inhibition of PARP, or the absence of PARP, you don't have this, and this question mark can be removed. You don't have this. You don't have this. So you don't have this septic shock. So PARP1 has a role in transcription regulation, specifically of these stress-induced transcription factors. So not only NF-kappa B, but also AP1. So transcriptional activation is also required, or for transcription, chromatin loosening is also necessary. Because if you have a very compact and dense chromatin, your transcription complex will not be able to perform its task. So this is why there's always a low level of PARP activity, because it's also involved in locally losing this chromatin, and for this function there's a very low activity of PARP1 required. So in case of PARP1 overactivation, there's this massive production of polymers, and not only to PARP1, it will also attach the polymers to all these other proteins of the transcription complex, which will lead to the complex will dissolve. It will not be a complex, but it will dissociate, and transcription is blocked. In the other part, the other way, if you inhibit PARP, this loosening of chromatin will also not take place. So this will also lead to a transcriptional block. So that is how PARP1 is connected to transcription. So how should you visualize this? So here you can see the NF-kappa B mediated transcription. So here you have P50, P65, NF-kappa B. PARP1 is connected. Not one molecule, usually there are more PARP1 molecules connected, but there is a function for PARP1 connected to the P300 CBP. This is the histone acetyltransferase. So PARP1 is necessary for histone acetylation. And here you can see it's also connected to another P50. It can also loosen the chromatin. So that is really what it does when it is active as a co-activator. So it mediates the recruitment of other activating proteins, and it also mediates the loosening of chromatin. So this already is clear, I understood. You have already had the acetylation, deacetylation as a transcription regulation process, so I'll skip this one. But there's another interesting connection with the histone acetylation and deacetylation. That is the sirtuin connection. Part 1 uses NAD plus as its substrate. And so do the sirtuins. They also use NAD plus and have ADP ribose to attach an acetyl group to. So if cells are under stress and PARP is activated, you will see this massive drop in NAD plus. And the NAD plus is no longer available for the sirtuins. So you will also see that the histone deacetylation by the sirtuins is inhibited to a great extent, which will also enhance cell death because P53 is acetylated when it's activated and when it's not deacetylated. So there is an enhanced induction of your cell death. So this is another way in which cell death pathway is well installed. So PARP activation can also indirectly enhance cell death pathways. So here you see all the possible roles PARP has in ROS-mediated tissue damage, oxidative stress, DNA damage, PARP1 activation. So that can lead to this NAD plus ATP depletion, necrosis, or to AIF release, which is here called apoptosis, but it's a caspase-independent apoptosis. And it also has a role in the activation of NF-kappa B, AP1, inflammatory mediator production, also cell death by DNA damage. So, and also, but that's less important, it plays a role in the induction of protein degradation. So if proteins get oxidized, they will be degraded, and PARP1 also plays a role there. So we, in our research, became also interested in PARP inhibitors because of this pathway. The activation of NF-kappa B is important in many diseases, degenerative diseases that we know today. So ischemia reperfusion is an important one, but also diabetes mellitus is associated with the activation of NF-kappa B, the production of INOS and superoxygen free radicals, DNA damage, and damage-induced necrosis and tissue damage. So PARP inhibitors play a role here, but also here. And if you have them active here, there's no need to have them active here because the damage will not be induced. So that was what we reasoned. If we know of PARP inhibitors that can help reduce this process, they won't be needed here. And especially if you have these chronic diseases with like diabetes, but we were also interested in chronic lung diseases, the reduction of NF-kappa B mediated gene expression may be very beneficial. And can we use PARP inhibitors for this? So these PARP inhibition has many applications, but there's a drawback, and I'll come back to that later. First, the ischemia reperfusion. So in heart ischemia reperfusion, you have the ischemia and reflow, which leads to a drop in ATP, but that's usually generated via the calcium levels, intracellular calcium levels that increase INOS production. peroxynitride DNA damage, and this is where PARP comes in. You will have PARP activation, NAD plus depletion, and ATP depletion. So you have massive tissue damage. So how did they study PARP inhibitors? It has very much been done in these kinds of models. You have an isolated heart in this Langendorf chamber, so there's medium flowing through the nutrients, and you can easily study what happens, at least by analyzing all kinds of mediators, proteins, products in this medium that flows through. So ischemia reperfusion is usually done by constricting one of these arteries and then releasing it again. And usually the restriction is 25 minutes, 30 minutes reperfusion, and then the measurements are done. So what you see here is normoxia, the vasoconstriction phase, and with the PARP inhibitor. So if you have normal oxygen, this is normal levels of NAD. You see here a drop in the constricted phase, and PARP inhibition, well, much of the NAD plus is still preserved. For ATP, the same. If you analyze all adenosine nucleotides, you see a similar pattern. And if you analyze lactate, like a measurement of lactate dehydrogenase, you see here that the constriction phase has a massive production of lactate, and it's also reduced when you have PARP inhibition. So we thought, well, we will test some natural compounds, so not in this ischemia reperfusion, but in other models. Why natural compounds? Because as part of Nutrien, the research institute you are interested in, can we come to some nutraceuticals that can be used chronically and it can be used in prevention of these chronic diseases? So we tested several natural compounds. We tested them in vitro with a purified, well, with the enzyme. It's a recombinant human enzyme, and it acts like normal PARP. We did it in cells. We measured the NAD plus levels, like I showed you before. We did it in vivo, in LPS-induced acute pulmonary inflammation, because we were interested mainly and collaborating mainly with the Department of Respiratory Medicine. And we did it ex vivo in human blood. And you can see they work very nice. As soon as you have discovered a compound that is a really inhibitor in vitro of PARP1, you can see all the expected features. So I'll show you. So we did this test and stopped it at 30 minutes and measured NAD levels. And we had a whole set of methylxanthines, but in fact these are caffeine and caffeine metabolites that were good inhibitors of PARP1. So not caffeine itself, that's this one. But major metabolite that is formed in our liver is this one, 1,7-dimethylxanthine. And also this one, 3 methyl and 1 methyl. So you can see two of the major metabolites that are formed out of caffeine have almost the same inhibiting potential as the classical PARP1 inhibitor 3-aminobenzamide. So what does this mean? Well, you can reduce necrosis induction. So we had some cells, we tested different cells, A549s and R24, which are endothelial cells. You can see that with 10 millimolar hydrogen peroxide, which is really high, so you completely, almost hydrolyze your cells, you can see there is a massive induction of ATP hydrolysis, so ADP is higher than ATP as long as it is over 1. So here you have necrosis and here you have apoptosis. And in the A541, you can really reduce the necrosis part to apoptosis with addition of this PARP1 inhibitor 1,7-dimethylxanthine. So this is also a measure of cell death. You can analyze the amount of ATP and ADP. So as long as the ratio of ATP over ADP is lower than 1, so ATP is higher, you have a normal condition like here, or apoptosis if there is cell death. And when this ratio is higher than 1, you have necrosis. So 17X shifted the cell death from necrosis to apoptosis, which is considered beneficial. So, next thing was this LPS-induced pulmonary inflammation. So we also compared this compound with threaminobenzoamide and with dexamethasone, which is a normal corticosteroid which is used a lot in inflammation as anti-inflammatory agent. So here you see after 4 hours and 24 hours the influx of myeloperoxidase or the myeloperoxidase that's present in the lungs, and this is a measure of neutrophil inflammation. So here the effect of the 17X, which is really reducing the neutrophil influx. So this is a normal condition. This is saline-treated mice, so these were just sham-treated, LPS-treated, and then also with 17X. 17X was just given through the mouth, so they had to swallow a lot of 17X, and then they had this LPS treatment. So it really seemed to work, even better than threaminobenzoamide or dexamethasone. So these caffeine metabolites clearly have an anti-inflammatory effect. It also reduced the level of IL-6, one of the pro-inflammatory cytokines, at least under these conditions. And, well, dexamethasone was just a little better, but it was really also an inflammatory treatment if you look at this aspect. What does it mean when you drink coffee, or take a Red Bull, or have something else that also has quite a high amount of caffeine? So we just wanted to know, what are we testing? What are we looking at? So here you can see the data of a very old study already that analyzed plasma levels of caffeine, of this 1,7-dimethylxanthine, and the other theobrom bromide, and some other theophylline after coffee consumption, of caffeine ingestion. And you can see the levels of this 1,7-dimethylxanthine are here 1 milligram per liter, here in the region of 3, 4, here also 4, here also like 3. So just to give you an impression, 2 milligrams per liter is like 11 millimolar in plasma. And that's something you reach with 2 cups of coffee. And this level will be above 1 or 2 for at least a period of, well, here, 10 hours, 12 hours. So with a few cups of coffee, you may have sufficient anti-inflammatory activity to expect some effects. That's what we tested. So what we did was we took blood of normal, healthy persons and of COPD patients. And this blood was tested ex vivo with LPS. We treated this blood with LPS, but then in presence of 10 micromolars of this 1,7-dimethylxanthine. So here you can see the levels of blood which was not tested in presence of this 17X, and this is what happens if you add this 17X. So in healthy controls, you could clearly see a drop in IL-6. This was statistically significant, only 10 persons. And TNF was also a little reduced in IL-8, and in COPD patients we only saw a clear reduction in TNF alpha, which of course for these patients is very nice if that happens. Not so much for IL-8, but IL-8 is very specific for, well, it's triggered to be at a higher transcription level in these patients. So that's no surprise that it's still a little higher. So, yes, at this level of 10 micromolar, it's really anti-inflammatory in humans. Oh, yeah. So, nice and interesting, and we also found some flavonoids that had some clear PARP inhibiting activity. So yes, you could use it in nutraceuticals to treat these chronic inflammatory diseases. You will may be able to reduce the plasma levels of pro-inflammatory cytokines, but there's another part of the story, which is the involvement of PARP1 or PARP activity in maintaining of the telomeres. The 10 kinases 1 and 2 are specifically active at telomeres, and there they have a very important role. But first, on oxidative stress and aging, you probably already had it in one of these lectures last week. Is that true? Did you already have something on telomeres and oxidative stress? No? Okay, that is different than last week. I'll explain. ROS DNA damage, that's clear. If there's no repair, or if when your antioxidant systems are not efficient enough, this will lead to cell DNA damage. The accumulation of damage will lead to cellular senescence, apoptosis. So if there's no tissue stem cell depletion, you may know that all tissues in our body, they have some sort of stem cells, progenitor cells, that are able to replace the damaged tissue cells. So if this happens to these cells, your tissue homeostasis will drop, and your organ function will decrease, and this will lead to aging. So PARP1 as a mediator of DNA repair. So if your PARP1 activity is good, it was thought, well, your DNA repair is sufficient, then this will prevent the accumulation of DNA damage and will enhance your probability to live a long life. And so some German researchers, the group of Alexander Dürke, tested the PARP activity in a specific white blood cell fraction, monocytes, and they compared this of all kinds of species and related it to the maximum lifespan of these species. So it was found that the higher the PARP activity, the longer the maximum lifespan. So a very nice positive association. So how about this telomere function? That was something we were also wondering with respect to this chronic application of our PARP inhibitors. Was that safe? Telomere shortening. So what you see in telomere shortening is that with cells that are deficient in telomerase, the telomeres will gradually decline, and this will lead to cellular senescence. But if there is telomerase, these telomeres will be more or less stable, at least they will not shorten that fast, and this will not lead to cellular senescence, it will lead to maintenance. In tumor cells, this telomere senescence induced by telomere shortening can be overcome, and then you have some sort of alternative pathway which also leads to telomere maintenance and cells that are able to proliferate. So telomere shortening is also thought to be a very beneficial effect, because short telomeres are also one of these mechanisms that prevent cells which an unstable genome from proliferating and eventually forming a tumor cell. So cell senescence is thought to be some sort of anti-tumor mechanism. And if the cell senescence is not working, or your cells are able to proliferate with a damaged genome, you will have tumors, and well, this is very bad. So telomerase is solving the end replication problem. For those that do not know, you have a leading and a lagging strand, and the problem arises in the lagging strand when the primer that is here at the end of the strand and is synthesizing the new DNA is removed, and this leaves a gap. And telomerase is overcoming, and if this gap is not filled, this will lead to a shorter telomere in the next cell. So telomerase adds some telomere sequence, and this leads to the synthesis of more telomeres because the primer can attach here, and you will have telomeres with almost the same length as the original cell. Part 1 has also a function in this telomerase system. So normally you have the telomeres, which are usually presented as a long stretch with repeating sequence of TTA-GGG, but they are formed in a specific loop, this T loop. And this prevents the ends of chromosomes, of two different chromosomes to fuse. So this is some protection mechanisms against chromosomal fusion, or fusion of an end of a chromosome to a DNA strand break. So this is very important in maintaining also the genomic stability. So there are all kinds of proteins also attached. So these proteins have got 1, TIM2, TRF1, TRF2. These are necessary to organize this T loop and to stabilize this loop. But if your telomeres have to be replicated, this loop has to be removed. And these proteins have to be removed because else your DNA polymerase cannot work. So I'll just skip this one. There is a role for PARP in this telomere stability, because PARP is necessary for these proteins, this TRF1, to be removed from the T loop. So the 10 chirases specifically attach polymers to this TRF1. So as long as this TRF1 is bound, telomerase cannot be active. If the TRF1 is removed via PARP activity, telomere elongation is possible, and the whole process of telomere stability is going on. So telomerase is active, new proteins are there, and telomerase is not active anymore. So PARP enzymes have a very clear role in this telomere elongation. So what we were interested in was whether PARP inhibitors, like the caffeine metabolites, were they good or bad? Was the reduction of oxidative stress and inflammation, was this beneficial in the long end because oxidative stress was prevented, and also the accumulation of oxidative DNA damage, and therefore with a chronic disease you may be able to live a longer and a healthier life? Or was this going on, like telomerase activity was not possible, your telomeres would shorten faster, and cellular senescence and organ function decrease would happen also faster? So we just did some very simple studies in this sense that we had a group of COPD patients which have a massive inflammation and oxidative stress. And we measured the telomere length in white blood cells. And we also tested this in healthy persons. So you have COPD and controls, and we just found that telomere length is reduced with increasing age, and it did so in both groups. So yes, telomere length is connected to aging. And coffee consumption, which we thought, well, that's a nice measure for the intake of caffeine, may be able to, via its anti-inflammatory effect, reduce the effect of aging and lead to longer telomeres. But we were not sure because, yeah, telomere length could also be compromised because caffeine metabolites would also inhibit this telomerase activity. But we found in both cases, both in healthy controls and in COPD patients, a mild positive association with coffee consumption. So that appears to be one of these, well, very mild indications. So I wouldn't see it's strong evidence or whatever. So it's just a suggestion that this anti-inflammatory effect may be more important than the effect on telomerase function. But in vitro, it's a little different. So if you have cells, you treat in vitro with a DNA damaging agent, in this case they used MNMG, and you do this in presence of PARP inhibitors, and not for a short period, so not for a few hours, not for one day. No, you have this inhibitor, the complete period present, so for 21 days, and you measure telomere length. What you see is that telomere length in these cells that were treated with highest dose of thioaminobenzoamide was shortest. So DNA damage or treating cells with PARP inhibitors for prolonged periods does lead to enhanced telomere shortening. But that's not so nice. So we thought we'd test also some of our PARP inhibitors in a similar system. That, I don't have the data here, but I can tell you that if we do the same experiment with one of our PARP inhibitors, we detected like the 17X, we saw a similar thing. So if you treat cells with this PARP inhibitor for 15 passages, you end up with shorter telomeres in these cells that were treated with PARP inhibitor than in cells that were not treated. So it's still one of these things that's puzzling us. Is it good? Is it bad? We're not sure. So this slide is some sort of recapitulation of everything I tried to explain. PARP1 is necessary for NF-kappa B and has a role in the induction of inflammation. It can cause NADP depletion, NAD plus depletion cell death, AIF release cell death, and this is important in ischemia, reperfusion, septic shock, Parkinson's, also type 1 diabetes. But also in neurodegenerative diseases, PARP1 is also heavily investigated at this moment. So also for those of you who are interested in in mental health and neuroscience as doing some sort of specialization, PARP1 is a hot topic there. And on the other side, it's also important in maintaining genomic stability, and that it does together with part 2 with the 10 kinases. It has a role in DNA repair, telomere maintenance, and genomic stability, and in this sense, it's also necessary to maintain a healthy genome and to live longer. So, all these things, again summarized, functions the facilitation of base excision repair, so specifically oxidative DNA damage, oxidized bases, cell death pathways, ROS-induced, transcription regulation, protein modification. So this poly-ADP ribosylation is what we call post-translational modification. Co-activator function and telomere stability, and many, because there are still some puzzling things we have not figured out yet, many other functions of part 1. Now, this is the applications of PARP inhibitors so far. So, ischemia reperfusion damage, this has been mainly done in animal models. In brain, it's a little funny, because you would suspect it would be beneficial there. And there are also some very clear data, because minocycline, the normal antibiotic minocycline, is one of the best PARP inhibitors that is known. We also tested it, and it appeared to be better than 3AB, better than one of these natural compounds we discovered. And there is also some literature from the neurosciences that says, well, if you have patients with stroke, treat them with minocycline, and they'll have a better perspective. So they will recover faster, their memory loss will be less, they will have a better recovery. Minocycline. It's also used to reduce vascular damage in diabetes. That has been a hot topic. It's less hot now because of these concerns about chronic application and genomic stability. Now, this chemo and radio potentiator, this synthetic lethality, is also a hot topic. Prevention of heart failure after ischemia reperfusion. It implies the chronic administration of PARP inhibitors, so it's still a little doubtful. Also, neurodegenerative diseases, Parkinson's, but also Alzheimer involve cell death. Still not clear whether you could use it chronically. And for these applications, these benefit risks evaluations have to be done. So there's no black and white statement you can make. So they're either good or bad. No, they may be better than the alternative, and in this case, you will choose a PARP inhibitor. Do you have any questions? If you have read the paper, do you have any questions on that already? No?